Okay, so today we're gonna teach this lecture over Zoom, and I hope everything works. Our topic today is about histone modification and chromatin accessibility. So we continue our epigenetic saga. In the last lecture, we talked about DNA methylation and the nucleosome positioning, right? We know that DNA methylation really silences the chromatin, and nucleosome positioning can be more dynamic, and very often transcription factors prefer to bind fairly nucleosome-free regions. The next part of epigenetics is histone modification. We mentioned that on this chromatin, the very basic unit is a nucleosome with histones, 8 histones, which form the nucleosome core, and the DNA wrap around the histone core. This will form the nucleosome. And you might notice on here, the histones are giving out these little protruding tails out there. These are called the histone tails. And they can be modified. And depending on the specific modification, the chromatin will have different properties. And so we look into this nucleosome in more detail. You can see here, there are very often, well, no, they're always 2 H2A histones and 2 H2B histones, 2 H3 histones and 2 H4s, and they are stacked on top of each other, right? And each of these histones can have a string of peptides shooting off from the histone. And this is the amino acid tail or the peptide tail of the histone. And those can be modified. And depending on the different enzyme, there are some that are histone writers, which basically can add more modification onto the tail. There are readers, which can sense, can detect whether there are modifications. There are also erasers that can remove the modification. And so depending on whether it's being added or removed, the histone tails will be different, and they have very different properties. And in order to know where these histones modifications happen across the chromatin, we can also again use ChIP-seq. And the experiment is similar to a transcription factor ChIP-seq. Basically, we still cross-link the chromatin to cross-link the protein that's and the DNA, right? So for transcription factor, it would mean a transcription, like for example, this would be a transcription factor that's binding to a nucleosome-free region. When you cross-link the transcription factor to the DNA. But in the cross-linking process, you also cross-link the histone proteins with the DNA, and then we can use sonication to fragment the chromatin into small pieces. And with transcription factor ChIP-seq, you use an antibody against the transcription factor of interest. But with histone modification, you can use different antibodies against a different histone modification. And this way you will be able to enrich for the whole histone or the nucleosome that has this modification, as well as the DNA that's attached to it. And that's how histone marked ChIP-seq work. Okay, so there are many types of histone modifications. And so this is an example, and this really tells us how you kind of call the histone modification. Most of the modifications that are very important, also available antibodies, are for the histone H3. Remember, there are 2 H3s, and its modification has the biggest effect. And depending on the amino acid location, so for example, this is a lysine 4, it will be H3 because it's a histone 3, lysine 4, and on this position, you can have methylation. And these are the methyltransferases. These are the writers that can add the methylation onto this histone lysine 4 location. And you can see it's lysine demethylation. If you look in here, it means histone lysine demethylation 5A, 5B, 5C, 5D. So when it's demethylation, it's an eraser. And when it's a black in here, you can see this is a writer. When it's, I think it's a yellow in here, it means it's acetylation. So these are different modifications. So by the way, acetylation happens only it's just one acetyl group, whereas methylation, because on each carbon, you have one that's linked to the remaining peptide, but then there are, on that carbon, there could be, one bond, second bond, and third bonds. And so the methylation can be monomethylation, dimethylation, and trimethylation, depending on how many of those bonds are methylated from a hydrogen to a methyl group. And so methylation can have, the reason we have so many, I guess, different methylation is some start from no methylation to a monomethylation, some will add a second, and some are specific adding to the third methylation mark on this location. And so you can see most of the marks are being added on either the lysine or the arginine locations. So you can either the R or a K, and that's where most of the acetylation, methylation happen. On phosphorylation, it's usually added to serine and threonine. You can see that's P, phosphorylation in here, okay? Most of the time when we talk about histone modification, we talk about either methylation or acetylation. And so there are some clarifications here. When we talk about DNA, it's just a naked DNA, double helix. There is no chromatin structure. Once we talk about chromatin, it means DNA in its compaction structure in the cells. And the nucleosome is the basic units of the chromatin, which includes the 8 histone protein, that's the histone core, as well as the DNA that's wrapped around it. And when we do transcription factor ChIP-seq, you use sonication, which is sonic wave to fragment the chromatin, and then you use the antibody against the transcription factor of interest. When you use histone modification ChIP-seq, by the way, you can fragment the chromatin either with just sonic wave, or if you remember the last lecture where we can use micrococcal nuclease, which is an enzyme that specifically cuts when there is no nucleosome, and they will chew the DNA until it reaches the nucleosome. And that will also basically give you all the beads on the chromatin, right? And so basically to shear the chromatin into small pieces, you can either use sonic wave or you can use the MNase enzyme to cut and chew. And here the antibody is not against the transcription factor, but against the specific histone modification on the nucleosome histone core. And the final result you get, for example, if we do just nucleosome sequencing, you will see that it really basically covers the whole genome. Everywhere it's covered, except that in locations where things don't matter so much, the nucleosome are not very well positioned. In places where this, say, if it's a beginning of a gene, you will have a more consistent nucleosome-free region, which is, like for example, in here, it's fairly nucleosome free. And also usually in the beginning of the gene, the nucleosome are better positioned. Whereas if you look at histone modification, this is an example where the chromatin is fragmented with a micrococcal nuclease. Basically, you can see most of the genome are not modified. Only occasionally those nucleosomes are being modified. In this case, it's H3K4 trimethylation. And you can see here, these are the nucleosomes that are enriched in the beginning of a gene. And very often, depending of course on the specific histone mark, it can cover 2% of the genome, 5% of the genome, and sometimes 15% of the genome. And so you don't really need to cover the whole genome over many, many times. If you do nucleosome sequencing, yes, you have to pretty much cover the whole genome. With histone modification, you also can see it's slightly better resolution and also better signal to noise. If there is, you can see basically it means that there is one very well positioned nucleosome, and this nucleosome actually aligns well with just the nucleosome sequencing above. And then the next nucleosome next to it is slightly less, modified with the H3K4 trimethylation, and the next one is even shorter, a smaller, lower level of methylation, and the next one will pretty much have no methylation. And this is, this one is the nucleosome-free region, which you can see also there is a big gap in here. Basically, because transcription start is there, the whole polymerase transcription initiation complex is there, and it squeezes out the nucleosome. And so you can see it has better resolution, better signal to noise. You capture the things that are potentially, you capture the nucleosome that have a flag in some sense, that are more interesting. And so over the years, people have looked at many different types of histone modifications. So there's two seminal studies where scientists use good antibodies to check each of the known histone modification with good antibodies to really characterize what type of histone modifications, what's their characteristic, where are they enriched in the genome. And so far from what people have learned is, for example, there is one or a couple of modification that really happens on the gene body. For example, H3K36 trimethylation, it really is enriched on the gene body, especially on the later half of the gene body, especially on the gene exons. And it is also kind of, so there are studies showing that, basically polymerase, when it sees the K36 trimethylation, it moves a little bit slower on the transcription. Whereas if there's no histone H3K36 trimethylation, for example, on the intron region, there's no K36, then polymerase can move fast because that region is gonna be spliced out intron, so it can move faster. If it makes a mistake, no big deal. And then when it hits a K36 trimethylation, you will know that the RNA needs to be correctly transcribed, and so it will slow down and do the transcription. And so K36 mark in the exons of transcribed genes are actually important for helping polymerase move on the gene. H3K79 trimethylation appears usually in the first exon and first intron regions. Another very important histone modification is H3K4 trimethylation. It appears at usually genes in the promoter of genes that are either active or poised for transcription activation. For example, in some cells, either this gene is expressed, then you will have K4 trimethylation at the promoter, or if the gene has a potential to be transcribed, but maybe it's waiting for some external signal to arrive, and so it's getting ready to be transcribed, it will also have a K4 trimethylation on the promoter. There are also important histone marks that happen on the enhancers. H3K4 monomethylation is a mark that's in the enhancer. It also is a little bit in the promoter because very often the promoter and enhancer actually interact with each other, and the enhancer will have higher K4 monomethylation, whereas the promoter will have higher K4 trimethylation. So you can imagine when the enhancer is looping to the promoter, the modifying enzymes is working very hard on the promoter to get things modified, and the enhancer will just kind of get a little bit of it. That's why it's kind of only having the H3K4 monomethylation. H3K27 acetylation is a very, very important histone mark. It tells you not only that this enhancer is important, actually K27 acetylation is also very enriched in the active genes in the promoter. Again, it's highly enriched at the promoter of the actively transcribed gene, and if an enhancer is also kind of looping to the promoter, there will be enhancers also having the K27 acetylation mark. So these two marks are very, very important landmarks across the genome, allowing transcription factors to find the place. As we mentioned, both, and also there are some marks that are enriched in both promoters and enhancers. For example, H3K4 dimethylation. Remember, we mentioned promoter has more K4 tri and enhancer has more monomethylation, but actually, depending on how things are close or far away, in both locations, you see enrichment of H3K4 dimethylation. And by the way, all of the acetylation marks are kind of enriched in both promoters and enhancers, and our ability to really investigate them really, I think, very often depending on the antibody, how specific and how good is the antibody. And the most useful one is this H3K27 acetylation histone mark. There are also marks like H2AZ. This is on the H2A. It's not really a modification, it's really a variant, which also kind of mark promoter and enhancers. There are also some regions that are more, a little bit repressive marks. So H3K27 trimethylation very often are enriched in genes that are silent, especially on the promoters of CPG island genes. If this gene is not expressed, usually you have a higher H3K27 trimethylation. H3K9 trimethylation usually appears in the repetitive regions of the genome. Usually this is a repressive mark that if this region needs to be silent temporarily, maybe you have a K9 trimethylation, but if it needs to be permanently silent in those repeat regions, DNA methylation will come over and cover those regions up. So these are repressive histone marks. And so knowing this, actually it's very, very useful to use histone modification to annotate the genome, to know, okay, this is the beginning of a gene, that's an enhancer, this is the active enhancer in the cell, and so on. And so we will have a lecture in the next, on Thursday, to actually teach you how to use histone modification and hidden Markov model to annotate different regions in the genome. Questions so far? I want to make sure that, you know, the students
I have a quick question.
Yes, hi.
How do you how do the peaks look differently between the different modifications? Are they all, do they have different, like how do the distribution of the peaks look?
Yeah, very good question. You can see a little bit of this from the slide 7 here. So for example, bisulfite sequencing is looking at the DNA methylation. You really have to cover the genome. You can see here, K27 acetylation give you pretty sharp peaks. K4 monomethylation occasionally give you a peak, but it's a little bit more diffused. On the enhancer regions, K4 monomethylation are a little bit more diffuse than K27 acetylation. K4 trimethylation is fairly enriched in the promoters, so it's pretty sharp. K36 trimethylation, because it's enriched in the gene body, especially in the exons, they are pretty broad. Also K27 trimethylation, because they appear either on the gene body of suppressed genes or on the CPG promoter of the CPG promoter is usually a few hundred base pairs, so it's also pretty broad. K9 trimethylation, because it's on the repeated regions of the genome, is very, very broad. And so the different marks definitely have a different property. Okay, so.